Assalamu alaikum dear students. Today we are going to discuss another topic of the developmental biology. This is the part 2 of the early embryonic development. The part 1 I will provide you in the description box. Please kindly watch that video along with this. Okay, let's discuss what the egg is. All the material necessary for the beginning of the growth and development must be stored in the mature egg, that is the ovum. The sperm has eliminated most of its cytoplasm, the developing yolk, for the oocyte before it reaches the stage of meiosis at which it is fertilized. Not only conserved its material but is also actively involved in accumulating more. The meiotic division that formed the oocyte conserved its cytoplasm rather than giving half of it away. The oocyte either synthesizes or absorbs protein such as yolk that act as a food reservoir for the developing embryo. Bird's eggs are enormous single cell, swollen with its accumulated yolk. Even eggs with relatively sparse yolk are comparatively large. Let's take an example, like the volume of the sea urchin. It is 200 picoliter, that is, it is 10,000 times the volume of the sperm. The sperm and the egg have equal haploid nuclear component. The egg has a remarkable cytoplasmic storehouse that it has accumulated during its maturation. Over here, there is a diagram of an egg. In the egg, there is outside membrane known as the jelly layer, and these are the sperms that are penetrating, right? You can see. Okay. There is a vitelline envelope. There is a plasma membrane, the cortical granules that are inside, the mitochondria which provide the energy, and the nucleus. The cytoplasmic trove includes the following. What are the things that are present inside the cytoplasm? That is the protein. The early embryonic cell need a supply of energy and amino acid. In many species, that is accomplished by accumulating yolk protein in the egg. Many of the yolk protein are made in other organs like liver and fat body and travel to the maternal blood to the egg. Okay, because for the egg we need supply of protein and it is transmitted from the maternal blood to the egg. There are ribosomes and the tRNA present in the cytoplasm. The early embryo need to make many of its own protein, and in some species there is a burst of a protein synthesis soon after fertilization. Protein synthesis is accomplished by ribosomes and tRNA. What is a tRNA? tRNA is a transfer RNA. What is its function? It transmits the RNA and it, in the process of DNA replication, you have often heard about the term replication, transcription and translation. The function of the tRNA is to transmit the RNA into a protein which exists in the egg. The developing egg has a special mechanism to synthesize the ribosomes, and certain amphibian oocytes produce as many as 10 raised to the power 12 ribosomes during its meiotic growth phase. What are the messenger RNA? In most organisms, the instruction for protein made during the early development are already packaged in the oocyte. It is estimated that the egg of the sea urchin contain 25 to 50,000 different types of mRNA. This mRNA, however, remains dormant until after fertilization. Okay. What are the morphogenic factors? Molecules that direct the differentiation of cells into certain cell types are present in the egg. They appear to be localized in different regions of the egg and become segregated What do you mean by segregated? Segregated means separated, individual separation into different cells during cleavage. Okay, before cleavage, all the cells are look like the same, but after the cleavage, the cell used to differentiate, and some of the cell becomes the liver or the heart or the skin or the outer membrane of the body or the brain, and so on and so forth. What are the protein chemicals? The embryo cannot run away from the predator and move to a safer environment, so it must come equipped to deal with threat. What is the threat? The threat from the predator. Many eggs contain ultraviolet filter and DNA repair system. Keep this thing very clear in your mind. Learn this. That protect them from sunlight. Some eggs contain molecules that potential predator find distasteful. What do you mean by distasteful? It means when the predator used to engulf or the eat, it tastes very bad. So nobody want to eat the bad or odor food like that. So this is also very beneficial. And the yolk of the bird egg contain antibodies. Okay, I'm talking about all these characters of the egg. These are very beneficial for it. Note it down. Within this enormous volume of the cytoplasm reside a large nucleus. In some species like sea urchin, the nucleus is already haploid at the time of fertilization. In other species, including many worms and most mammals, the egg nucleus is still diploid and the sperm enters before the meiotic division. What are the stages of the egg nucleus at the time of sperm entry in the different species is given below. Okay, this is the primary oocyte and you can see there is a germinal vesicle. Okay, this is the first metaphase, this is the second metaphase, and this is the meiosis. And the meiosis, it contains the polar bodies and the female pronucleus. We are going to see what are the stages of the eggs nucleus at the time of sperm entry. Okay, let's take a look. Enclosing the cytoplasm in the egg plasma membrane, this membrane must regulate the flow of certain ions during fertilization and must be capable of focusing with sperm plasma membrane. Outside the plasma membrane is the vitelline envelope which form a fibrous mat around the egg. This envelope contains at least 8 different glycoproteins and is often involved in sperm egg recognition. Okay, in the fourth point, learn and keep this in mind that at least 8 different glycoproteins and is often involved in sperm egg recognition. Okay. The fifth point, it is supplemented by the extension of the membrane glycoprotein from the plasma membrane and the proteinaceous vitelline pores that adheres the vitelline envelope to the membrane. The vitelline envelope is essential for species-specific binding of the sperm. Species-specific binding of the sperm. In mammal, the vitelline envelope is a separate and thick extracellular matrix called the zona pellucida. Okay. The mammalian egg is also surrounded by a layer of cell called the cumulus, and which is made up of an ovarian follicular cell that were nurturing the egg at the time of its release from the ovary. This is the function. Okay. The mammalian sperm have to get past these cells to fertilize the egg. The innermost layer of the cumulus cell immediately adjacent to the zona pellucida is the corona radiata. Okay, this is the plasma membrane and these are the microvilli. This is the vitelline envelope and these are the cortical granules. Lying immediately beneath the plasma membrane of the egg is a thin shell of a gel-like cytoplasm known as the cortex. The cortex in this region is stiffer than the internal cytoplasm and contain high concentration of globular actin molecules. During fertilization, these actin molecules polymerize to form a long cable of actin known as microfilaments. Microfilaments are necessary for cell division and they also used to extend the egg surface into small projections called microvilli, which I have shown you in the previous diagram, which may aid sperm to enter into the cell. Also within the cortex are the cortical granules. These are the membrane-bound structure which are homologous to the acrosomal vesicle of the sperm. What are the acrosomal vesicle? I have already explained it to you in the previous lecture. Please kindly look at that. Are Golgi-derived organelles containing the proteolytic enzyme. Each sperm contains one acrosomal vesicle. Each sea urchin contains approximately 15,000 cortical granules. Moreover, in addition to digestive enzymes, the cortical granule contain mucopolysaccharides, adhesive glycoprotein and hyaline protein. The enzymes and the mucopolysaccharides are active in preventing other sperm from entering the egg after the first sperm has entered, and the hyaline and the adhesive glycoproteins surround the early embryo and provide support for the cleavage stage, the blastomere. This is the structure of an egg. You can see there is a cumulus, the ovum is present and the zona pellucida. Okay, this is the diagram. Note, many types of egg also have an egg jelly outside the vitelline envelope. Egg jelly, it is mainly present in the amphibian. You can see when you look at the egg. Okay, many glycoprotein meshwork can have numerous functions, but most commonly is used either to attract or to activate the sperm. The egg then is a specialized cell for receiving sperm and initiating development. How we recognize the egg and the sperm? The recognition of egg and sperm. The chemoattraction of the sperm to the egg by soluble molecules secreted by the egg. The exocytosis of the acrosomal vesicle to release its enzyme. The binding of the sperm to the extracellular envelope, the vitelline layer or the zona pellucida of the egg. The passing of the sperm through this extracellular envelope. Fusion of egg and sperm cell, the plasma membrane. Sometimes the step 2 and 3, these 2 points, are reversed as in mammalian fertilization, and the sperm bind to the egg before releasing the content of the acrosome. Okay, this is a diagram of the sea urchin and this is the mouse which shows the sperm contact. You can see the acrosomal reaction, digestion of the jelly, binding of vitelline envelope, the fusion of acrosomal, the 5th point, the fusion of acrosomal process membrane and the egg membrane, and this is the plasma membrane. But in the mouse you can see the sperm activated by female reproductive tract. Okay, this is the flagellum, this is the nucleus, this is the acrosome. But in this it is only has a nucleus, the centriole and the actin and the acrosome. Okay, the sperm binds zona pellucida. Okay, but over here the acrosomal reaction directly takes place. The third is acrosomal reaction. You can see over here the digestion of jelly layer takes place. The fourth is sperm lies whole in the zona pellucida. Okay, and the 5th is the sperm and the egg membrane fuse together. This is the outer membrane is the cumulus layer, that is the extracellular core, and this is the zona pellucida, the extracellular matrix. This is the egg cell membrane outside it. Okay, After these 5 steps are accomplished, the haploid sperm and the egg nuclei can meet, and the reaction that initiates the development can begin. Okay? With the help of diagram, you can discriminate. Note down a point. In many species, the meeting of the sperm and the egg is not a simple matter. Many marine organisms release their gametes into the environment. That environment may be as small as a tide pool or as large as an ocean. Moreover, it is shared with other species that may shed their sex cell at the same time. What is the question that came into the mind? These organisms face two problems. We have talked about how the sperm and the egg meet in such a dilute concentration, and how can sperm be prevented from trying to fertilize the egg of another species. The answer is that two major mechanisms have involved to solve this problem. First is the species-specific attraction of an sperm. What do you mean by species-specific? Means the Homo sapiens, like the humans. The sperm of the human male can fertilize the egg of human female, Homo sapien. It does not mean that the sperm of a human fertilizes the egg of a frog, female egg. And the species-specific, like we are Homo sapiens sapien, right? Species-specific sperm activation. Okay. Thank you.